Thank you. It's a privilege to be here at the ISMB and getting to talk a little bit about our work for enhancing AI readiness of the gnomAD resource using the GA4GH Genomic knowledge standards. For those of you that are new to the area of genomic medicine, just in general, the principle is we get patient or tumor DNA. We want to figure out what changes in that DNA are driving potential clinical action, and then report that back to help influence patient care. There are a number of standards and methodologies used to help in the process of interpreting patient genomes. The one shown on the screen here are the ACMG guidelines that are well known and established for determining the pathogenicity of variants in constitutional disorders. Notably, in these guidelines, there are a number of different types of evidence that you might use in order to drive your interpretation of a variant towards pathogenic or benign, as shown in this table. Each row in this table is a different type of evidence, and each column represents the growing strength of evidence from either strongly leaning towards benign all the way to strongly leaning towards pathogenic. And the idea here is that by interpreting the evidence you observe in a variety of different contexts, applying codes to it, these codes are shown in this matrix, things like BS2, PS4. Using the rubric shown on the right, you're able to combine these codes in order to come up with an overall classification for the pathogenicity of a variant. Now, in order to do this, our frustrated variant scientists need to go to a variety of different evidence resources, collect that evidence, encode it using this framework, and then ultimately use that to drive a classification. Because there are so many resources and there's a growing number all the time, there's this effort to try and harmonize all the evidence that exists across databases inside centralized data warehouses shown here. Unfortunately, this leaves our variant scientists now traveling to various data warehouses and trying to look through all of the evidence that's supplied between them, which is more or less non-overlapping. So what we're trying to work towards is a future where all of these different resources all speak the same standardized language, where evidence is all structured and allows for interoperability between these resources, allowing for scalable AI applications and clinical decision support. We're doing this under the context of the Global Alliance for Genomics and Health. So for those of you that are unfamiliar with this organization, the GA4GH is a standards development organization. What they do is they work together to create policy framing and technical standards in order to drive genomics and health sharing under a human rights framework. They do this for a number of different standards that many people in this room are probably familiar with. For example, they develop and maintain the VCF, the BAM and SAM standards, CRAM, bed files, etc. And the way they work is that they have these 6 technical and 2 foundational work streams. These are worked on across a number of real-world driver projects that together are used to drive the development and maintenance of these specifications. Today, we're going to be talking about the activities and standards developed by one particular work stream, the Genomic Knowledge Standards work stream, and the driver projects that are primarily driving the standards development in this space are the Variant Interpretation for Cancer Consortium and the ClinGen driver project. And all of that work is currently being done through this R35 mechanism from the National Human Genome Research Institute to build a framework under which we can associate evidence with variants in order to create these assertions of pathogenicity, oncogenicity, therapeutic response, et cetera. And under the NIH ODSS supplement, we were then working with the gnomAD resource, collaborating with them in order to apply these standards to that resource at scale. gnomAD is very helpful, looking back at our ACMG framework, for determining population data. So it's a very large aggregative genomics, it's called the Genome Aggregation Database. It does exactly what it sounds like. And it allows us to look at the frequency of variants that exist across various human populations. And so what we've been doing is we've been working on developing a cohort allele frequency model that allows us to take the variants that we observe and basically represent them using these GA4GH standards and associate that with a standard for cohort allele frequency evidence itself, so that when we have a statement like this, Brugada variants classified as benign for Brugada syndrome, which is supported by standalone benign evidence from the ACMG framework, based on a gnomAD genomic ancestry group allele frequency of 0.083, that we can structure all of that so that a computer system can then take these individual components and reinterpret them, integrate them with other evidence, and do it all automatically, improving the scalability and helping our frustrated variant scientists. So to do this, we developed a CAF schema, a cohort allele frequency schema, under the GA4GH variant annotation specification, and we applied the pre-existing GA4GH verse specification for variant representation. So a bit about gnomAD. gnomAD is a very large aggregative database of genomes and exomes, and these come from a variety of different resources. Shown here are all the different projects associated with it. And it's well over 100 studies, over 300 data contributors across 25 different countries. And in total, it's over 730,000 exomes and genomes. So it's quite large. There's a lot of data in here, and that helps us think about how do we apply these standards at scale. So one of the standards we're using is the variation representation specification. This GA4GH specification helps us with how we do interoperable exchange of variant data such that it's machine readable and interoperable across resources. It's based off of a core terminology and information model that defines all the different components: what's an allele, what's a sequence location, what's a sequence. And on top of that, it builds a JSON schema. There are GA4GH conventions for how we normalize variants using this specification, and then a reference implementation that takes these and applies it so that you can then use that in an applied setting. And what this allows us to do is create interoperability across a variety of different formats: protobuf, JSON schema, XML. Makes all variants self-descriptive, where it's effectively an additive collection of building blocks that allows computers to understand all the components used to describe a diversity of variation concepts. And most importantly, it's globally consistent and decentralized. So you can have the same variant occurring on sequences with different names that resolves to the same digest if it represents the same change on the same underlying data. And this allows us to create basically a global network of federated identification without any prior coordination. And so in order to do this in gnomAD, we had to think about how we do this at scale. Of course, this is a big challenge. There are almost 1.9 billion variants in gnomAD. For this, we created some high throughput annotation tools. We were able to extract the entire GVCF from the gnomAD resource, annotate it, and then re-import it. And this exists now on the gnomAD v4 data release. Finally, just a few words about the cohort allele frequency model itself. This is a model that allows us to create a representation of the frequency of a variant that exists in a population. More importantly, it allows us to look at subpopulations recursively. So you have your general population frequency, you have subpopulations with different genetic ancestry groups. You also have subpopulations within those subpopulations of different, biological sex, for example. And in addition to this, we also had to build standards that allowed us to create kind of resource-specific quality metrics and ancillary results in a standardized way. And so all of this is incorporated into the CAF model. There's not really time to get into any of the details here, but if people are interested, there are links in the slide. Okay, so I know it's a short talk. There's a lot of data there. So if you have any questions, of course, feel free to come up to the mic or shoot me an email. It's right up here. The slides are available online, so you can go and you can look at these and you can click through the links and find more stuff about it. You can go to go.osu.edu/ISMB-gnomad-GKS. So if you want to find out more about it, you can find these slides online at that URL. Okay, with that, I'd just like to acknowledge my team. I'd like to acknowledge the team at the Broad Institute at gnomAD. And then finally, of course, both the NHGRI as well as the ODSS for supporting this opportunity. And with that, I'm happy to take questions.
One quick question. So how do you integrate quality of variants into this automated way? Because when genetic counselor review, they look at more depth in terms of who deposited the data, and it was the older data, and now there is more better technology. So they have a lot of subjective measures that they put into the variant annotation and getting that information out?
That's an excellent question. So what wasn't shown here, and just for purposes of time, is that the variant annotation specification has a rich evidence and provenance model around it. So you're able to basically build in the different actions taken in order to generate the evidence that was used. This includes not only where it came from, but also if there's any like human or automated interventions, the notion about what those are, what actions were taken in order to create that data. And because all of that evidence and provenance is attached to each individual data item, then you can integrate across multiple institutions while preserving all of that rich history, allowing people to do advanced analytics as to, consistency of quality, as well as reliability of data that comes from different institutions and individuals, if that's of concern to that institute.
Thank you very much. Any other question? A quick one? Yeah.
Thank you, Alex. I know the incredible amount of work that this represents, and I am wondering if you can share a little insight regarding the requirements you've captured from the community for the development of the standard, and how do you see the community adopting downstream? Just curious to see that.
Yeah, absolutely. So as I mentioned earlier in the talk, the GA4GH is this intersection between technical work streams and driver projects. And as part of the process of developing these specifications, it's important that we work with driver projects to basically outline what their use cases are. For our specific purposes, of course, there are a variety of different kind of end of the road knowledge bases that describe variant interpretation data. I think one of the best known is ClinVar, which is hosted by the NCBI. If anybody ever looks at the ClinVar statistics page, you'll find something striking, which is that over 20%, I think it's close to like 23% of all multi-submitter classifications are conflicting. And there's no way to resolve this. You have to do it manually. You basically have to go in, you've got to recontact the labs. You have to get their information. Why do you report it this way? And then a human has to go and integrate all that and report it. And the reason why is because the ClinVar data model does not support kind of this rich structure of interpretation down through evidence. And so this is like one of those driving use cases. We're trying to advance the field to the point where you can actually have multiple submitters using that same data structure so that resources like ClinVar can then actually computationally integrate, reinterpret, and provide consensus interpretations.